Dear students, now I am going to discuss about the topic fluorescence activated cell sorter, that is FACS. So FACS is a particularly a technique that can be applied in different field of biological sciences, mostly for cell sorting, means from the mixture of the cells you can identify and also sort a particular type of cells, and also you can enumerate, means the percentage of cells that particular cell is present in the mixture. You can get the idea of that. So what is FACS actually? Before going to discuss about FACS, I'd like to discuss about flow cytometry, because flow cytometry and FACS, they are very much similar. They have very much similar applications. The two type of instruments, more or less similar, but some additional attachment is there in FACS rather than in flow cytometry. But the principle is almost similar. So here from the term flow and cytometry, so this particular wordings there, it is showing that flow means something that is in motion. Cyto, that refers to cell, and metry means measure. So using this particular technique or technology, what we are measuring? We are measuring cells that are in motion. So you may have some question in your mind that already we have developed electron microscopy, and by using electron microscopy, irrespective of same or TEM, in scanning electron microscopy or transmission electron microscopy, we can get the ultrastructural view of a particular cell. Then what is the need for this particular kind of or additional kind of technology? So the development of this particular instrument for the application of mostly application in case of cell biology, cancer biology, we need this instrument because in microscopy, and if we have selected a particular field, and that particular field you can have different types of cells. So you cannot segregate, you can separate all the type of cells, and you cannot confer that, okay, this is the type of cell that is only present in my sample. So this is a means we are getting a picture, maybe an ultrastructural view of cells. But to get a particular form of cells, to get a particular type of cell, same or same is not a handy instrument. And also the cell to be fixed, so it's not in its actual condition that we are detecting. So it's in freeze condition or fixed condition. So it may lose some of its property during fixing or the freezing condition. So here is the importance of this particular instrument for the application of cell biology and cancer biology. So mostly this particular instrument was proposed to use no, for measuring the properties of single cells in a fluidic system. And first flow cytometry device was discovered in US in 1953 by W.H. Coulter. That is why this instrument can also be called as Coulter counter. Then that was a flow cytometry instrument, not the FACS one. The FACS was first invented by the company is known as Becton Dickinson BD in 1974. So this particular instrument gives us the ability to analyze many properties of many cells within a very short span of time. And for routine use in case in the diagnostic laboratory to diagnose any kind of health disorders, mostly focusing on the cell types, and to detect blood cancer, this instrument is prerequisite in the diagnostic laboratory. But this particular instrument has many other applications in basic research, in clinical practice, in clinical trials also. So for the purification system, or to identify antibodies present in a particular type of cells, you should use this particular instrument for the identification from a different population of the cell types. So flow cytometry, if I try to define flow cytometry, so the definition would be like that in biotechnology, flow cytometry is a laser-based biophysical technique employed for counting cell, for sorting cell, and to detect biomarker and protein engineering by suspending cells in a stream of fluid and passing them by an electronic detection apparatus. So it allows simultaneous multi-parametric analysis of the physical and chemical characteristics up to thousands of particles per second. So this is the novelty of this particular instrument, that you can simultaneously analyze multiple parameters belongs to physical or chemical characteristics of a cell, not a single type of cell, but thousands of cells per second. So different parameters of thousands of cells can be measured within a second using this particular instrument. Now I'm coming into the basic component of FACS. So FACS contains mostly three different components. One is the measuring system, one is the sorting system, and the third one is the analyzing system. So the basic concept of FACS is that it measures the cells that is under the fluidic conditions, that is in motion or in flow system, and delivers the cell singly past a point of measurement. And the light is focused. So the cell is in motion. So within the instant of time, what is the condition of the cell? So we can measure it. Then this particular instrument enables sorting of a population of cells into subpopulations. Instead, assume that 10 to the power 6 number of cells are there. It may be of 4 to 5 different types, and within few seconds, we can sort out that how many type of cells are there in say in group 1, in group 2, group 3, and group 4. If there are 4 different type of cells present there in the mixed population. So it's not a manual counting, it's an automatic electronic counting. Or through automation, we can count it within a few seconds. And also we can sort it. But what are the percentage of, say, group A type of cell is present there? So this is very effective. Very specifically, we can do this cell sorting because here we are using an antibody-based technique, and we know that antibodies are very specific protein that should be coated with a signature molecule. Mostly these are the fluorophore or the fluorescent dye, and this fluorescent dye will gives us the data, the indication. As because the antibodies are very specific, so they will bind with the specific type of cells, and then if we illuminate the fluorescent dye or the fluorophore that is tagged with the antibody, they will illuminate and we can get the data. That is why this process is very effective one. So to carry on this particular instrument, we need 5 different things. The first thing is the single cell suspension of antigen, of the antigen. Next one is an antibody conjugate. Third one is the fluorophore or fluorochrome and fluorescence. Fourth one is the flow cytometer, and the fifth one is the analyzing software. So these are the 5 different components of facts that we need to carry on this particular test. And now, what are the steps? How to carry on this process? So step 1, that is preparation of cell. So you have to prepare the shell as such that there will be no clumping. That we have to take care. That there should not be clumping of cells. Next one is the staining one. So we have to stain the cell using the labeled antibody, means the fluorophore tagged antibody with the cell. Next part is next step is cell acquisition. Means we have to perform the experiment. Then we will go for cell analysis and data interpretation. So these are the 5 steps for flow cytometry analysis. Now, as I said, the flow cytometry and FACS, they are very much similar in type of instrument, but they have some differences also. The similarity is that in both the cases, we have to add fluorescent tagged antibody to detect the cell type. And this particular instrument should be used only for cellular analysis, not for the other purposes. For cellular analysis or cell level analysis. What are the differences? Mostly flow cytometry commonly found in research lab. This is very user friendly, but FACS is a relatively costlier instrument. Personalized research lab we have, and mostly this is designed for research purposes for the researchers, and application of FACS is a very complicated one. A novice person cannot operate FACS and also cannot interpret the data. Next one, flow cytometry cannot sort cells, but FACS can. It can sort a heterogeneous mixture of cells into different populations. Then intuitive software interface is there, but here in case of FACS, this is automated. Everything you will get through automation, so you don't have to do anything by manual. So the principle of FACS or flow cytometry is that the passage of cells in single line in front of a laser so that they can be detected, counted, and sorted. So if you look here in this picture, you can see this is a nozzle. And this particular nozzle that is dispensing the sample, and now gradually the cells are coming through this nozzle one by one. It's not that multiple number of cells are coming and hitting the laser beam. Here is a laser beam. This is the laser beam. Here you can see this is the laser beam. It's not that at a particular time only one cell is hitting the laser. Not that the agglomerate is why I have mentioned that cell preparation is a tricky one. You have to prepare cells like that. There should not be clumping of cells. So one by one, cells are passing through this fine nozzle, fine tube, and hitting with the laser beam here. And there we can measure the two types of scattering, forward scattering and reverse scattering. I will come into that part. So here you can see now we have two types of detector. So they will detect the signal that is coming from the cell, and that signal should be analyzed using a software in computer monitor. So this is the basic. So from that, you can say that we can have three different components. Here, one is the fluidic component. This one is the fluidics. It's called the fluidics or the fluid flow system. So one by one, the cells are coming and hitting with the laser beam. And here, this is the second one, that is the laser section. We have different kind of laser with different intensity, with different wavelength of light. So this is very light sensitive. So this is called the optics system or the optical system or optics. And this part is the analyzing part. This is the electronic system. So that is actually processing the signal coming to the detector. So basically, flow cytometry or FACS, they have this basic three component. But in FACS, there is additional component, that is the cell sorter. So the fluidics, here you can see the fluidics, they have the cells are like here, and here we have to add a particular fluid that may be buffer, that may be seed fluid to maintain the fluidity of the particle or the cell. So the fluidic system is used to transfer particle from a randomly three-dimensional in orderly system of particles. And here you can see that in the optic system, it is composed of 5 different subcomponents or subsystems. So the first one is an excited source, next excitation source. The next one is a collection optics, then steering optics, filter optics, and PMT, photomultiplicity. So optics are central for flow cytometry for the illumination of stained and unstained particles. So those particles that are unstained means not tagged with antibody, not attached with antibody, they will not illuminate. So for the detection of scatter and fluorescent light signal, we can use this optic system. And this is where the particles the cells are passed through at least one laser beam. So there are 4 laser beams, so at least one laser beam they should pass. So lasers are available at, as I said, lasers are available at different wavelengths ranging from ultraviolet to red, from far red. So mostly the commonly used lamps are mercury lamp or xenon lamp. Now high power watercolor lasers, say argon lamp, krypton lamp, dye laser. Then radium, then helium neon laser also available. So they are, here you can see, these are the lasers that can be used in flow cytometry or FACS. So mostly they are used to result the light signal. And this light signal will develop some electronic system. That is the electronic system that is getting the light signal through the detector And then there is an amplifier. There are 5 different type of detectors: fluorescence signal 1, 2, 3, and then side scattering and forward scattering. So here you can see the type of detectors. Here is a type of detectors. So the signal that is coming from this optical filters, there are 5 different optical filters also. Here is a 5 and here is a 4 different optical filters. So when this is coming to the detector, then it will pass through an amplifier, because the signals are very faint, very less. So this have to amplify. So there is an amplifier that is actually amplifying the signal coming to this detectors. There is a forward scatter detector, side scatter detector, or the fluorescence type of fluorescence detector. Then there is a conversion of digital conversion, from analog to digital conversion of your signal, what you are getting through the detector, and it will come into your computer panel, a monitor. Now you have to analyze it using some compatible software. So as this picture says, so this x-axis says forward scattering of the cells, and the y-axis says side scattering of the cell. So here we are getting different dense, different region of cells. These dots are saying the population of cells. And you can clearly see here, you can see that, here you can see that there are 3 different populations of cells, maybe 4 different populations of cells. So that part I'm coming. First, this is how the experiment is going on. Here is a mixture of cells. First part, here you can see mixture of cells. Now one by one cells are passing through and then hitting with the laser beam. There is a photomultiplier tube that is actually collecting it and amplifying it and then delivering it to the computer. Now you have different type of filters: green filter, then red filter, for red filter, blue filter is also there. And in case of FACS, a sorter is there, but the sorter is absent in flow cytometry. Here you can see the additional attachment that we haven't seen in flow cytometry here. So this is an electrical attachment that is actually imparting some charge to the cell type, and then this cells has been collected in different collector. This is called a fraction collector. So here you can see the fraction collector. So those who are the positive type of cells are having positive charge on the surface, they'll be collected here, the green one, and no charge, the middle one, the gray one, and negative charge with the positive one, the red one. So this is the attachment that is present there in FACS and not present in flow cytometry. So cell sorter is there that can sort the type of cells and collect it in different collector, fraction collector. So here is a process, detailed process that I have already discussed. I'm not repeating it. And this is how forward scatter and side scatter is happening. So forward scatter means light scatter at a degree of the axis of laser path is referred to side scatter. And here you can see that in all the direction that is coming, it's not at a particular direction the scattering light is coming. But in this case, forward scatter, only this particular direction is forward scattering. Here, in this case, the light is coming. Here there is forward scatter as well as the side scatter. So there are 2 detectors that is actually detecting the one is detecting the forward scatter, one is detecting the side scatter, and the other 3 that are detecting the fluorescence signal. So on that basis, we can have 3 different populations. So this is the case of performing flow cytometry. FACS or fluorescence activated cell sorter using lymphocytes. So lymphocytes of the cells, monocytes of the cells, neutrophils of the cells. So I'm taking blood cells, a mixture of blood cells. Now I have tagged these blood cells with fluorescence conjugated or fluorescence probe conjugated antibody. And now I'm getting three different populations here. This is forward scattering along x-axis, and y-axis there is side scattering. And here you can see this dense population. Here these are the lymphocytes, that are smaller cells, so low forward scattering with few granules are present there. So this is low side scattering. In case of neutrophils, they are large cells, so high forward scattering. Here you can see more high forward scattering as well as high side scattering. In case of monocytes, they are large cells, so high forward scattering, but few granules are present there, so very few, when it's very low side scattering. So side scattering can be occurs if they are granular. Forward scattering will be occur more if their size is more. Size is big. So small type of particles, small type of cells that are not much of granular, there will be less side scattering, there will be less forward scattering. So large type of cells, there will be more forward scattering. And if they are granular, then there will be more side scattering. If not, then there will be less side scattering. So we can interpret the type of cells we have, and also we can enumerate the percentage. How much percentage of lymphocytes, neutrophils, and monocytes are present there in your provided sample. So by using this technique, you can separate out, and you can also get the percentage of type of cells present there in your sample. So this is how you can perform your test. These are the collection tube. This is the waste. That is not at all bound with the antibody we have already added. So the fluidic system maintains the flow of the cell and also guiding the cells to come one by one and hitting the laser beam. So the vibrating nozzle is there. This is the vibrating nozzle. So that is actually responsible for the flow or the motion of the cell. Then the laser, this is the laser. Here you can see the laser source that is actually focusing the laser and hitting the cells. There may be two type of scattering, forward and side scattering, and then fluorescence. And that should be detected using some collection optics as well as filters and amplifiers. So they will amplify the signal that is coming through detector, and then the electronic signal on the computer system that is interfaced with the software, and that software will interpret your data and will tell you about the sample composition. So here two type of antibody can be used, but that antibody should be tagged with fluorescent probe. So the antibodies may be monoclonal antibody, maybe polyclonal antibody. So monoclonal antibody application is more preferable because they are more specific. So they have the specific epitope, and they will bind with a specific type of antigen. But if not available, we can use polyclonal antibody because immunochemically dissimilar antibodies and react with various epitopes of given antigen. So it's better to use monoclonal antibody in case of flow cytometry of FACS. And here are the fluorophore we can use. There are different type of fluorophore can be used. Say fluorescein isothiocyanate is a green color fluorophore. It has the excitation 488 and the emission 520. Then phycoerythrin, energy coupled dye. Then here also phycocyanin, then propidium iodide, PI you can use, and then phycoerythrin, cyanin 5, PC5 you can also use. There are some other kind of fluorophores also there, and their respective excitation wavelength and emission wavelength is listed here. So we can use mostly the red one and the green one is used, as well as along with the blue one. Blue one is a DAPI, that is to stain the nucleus only, but mostly to target the antigen as well as the antigens, that is intracellular or the surface. So we have to add the fluorophore tagged with antibody, and here the fluorophore mostly used is fluorescein isothiocyanate and rhodamine or else phycoerythrin, phycocyanin. These are the detectors. Not only in the dot plot, we can also get it using this Gaussian curve. Also, you can get Gaussian curve, and we have to interpret there is an interface of software is there, and the software will interpret what is the type of cells. And if we are here, the example is given here is a type of different type of cells. We have taken blood cells, and there you can interpret by analyzing this data that how much percentage of lymphocyte monocytes or neutrophil are present in your blood sample. This is how you can detect. You can also detect it, use it for apoptosis study. So these are the cells that are not bound with any kind of antibodies. So these are the normal cells. These are the cells means this bound with the green antibody means FITC. So if there are primary apoptosis, so there will be more forward scattering. If there are secondary apoptosis, then we can get cells here. So here means the cells are going to be die. So both the red and the green, both will bind with, both the types of antibody will bind with the cells, and we can get the cell population here in this third compartment. And the fourth compartment, if we get large number of cells, then this is on for side scattering is red, that is bind with red, means this has already the cells are dead. So that is how by interpreting the data, we can convert that how much amount of cells are in normal cells, how much are primary apoptosis, or how much are secondary apoptosis, and how much is dead cells. So this is the application of flow cytometry. This can be mostly used for clinical use. So for genetic disease detection, DNA analysis in case of malignancy, then cell types in case of apoptosis study, in case of hematology, immunophenotyping applications we can use, in case of enzymatic deficiency studies, and in case of leukocyte analysis, we can use flow cytometry. Here are the references. You can get so much of data from these references. Thank you.